Forgot to record, so this is a re-recording. Alright, so we talked about plants and nutrients in this recording. And we started by talking about how nutrients can be moved throughout the plant, right? And nutrients and sugars are moved through the sugar-conducting cells, which we have already seen as phloem, right? Now phloem, if we look at the very specific cell type that makes up phloem, that would be our sieve tube elements. Right, so that they're the very specific cell type. They are these long cells that we see in our image over here. Right? They are actually alive at functional maturity. And you wouldn't think I would have to specify that, but xylem, which conducts a lot of the water, is actually dead at functional maturity. Right, so sieve tube elements, they are alive, but they lack most of their insides. So they lack most of the organelles. They don't even have a nucleus, right? Most cells will have a nucleus that contains their DNA. These cells do not. So they have to rely on companion cells, right? And these companion cells will act kind of as like the controller for these sieve tube elements. They contain nucleus, they contain other organelles, they serve those sieve tube elements. Now the importance of phloem. Well, phloem transports stuff, right? What is that stuff? Well, it's the transport of sugar, nutrients, and other molecules between where they are made and where they are needed. And we're really talking about long distance transport here, right? So transport throughout the entire plant rather than short distances, which would be like cell to cell, right? They can also transport a lot of regulatory molecules like different hormones. They'll also transport water and help redistribute water throughout the plant as well. Now, the actual term for the transportation of these sugars and other types of molecules is translocation, right? So that's the process of actually moving. I'm going to stick with saying sugars, right? Because a lot of what is moved is sugars. The process of transporting sugars to where they are needed in the plant. And so see in this image over here, we have our leaf cell, which is actively producing sugars, right? The sugars will be loaded into the companion cells, which then helps the sugars being to be loaded, into the sieve tube elements, right? They'll then travel throughout the plant and then leave the phloem and enter into the cells that need the sucrose. You'll also see on this image I have a picture of the xylem. The xylem works in tandem with the phloem as it provides a lot of water to enter into the phloem and exit as well. So why do these sugars even need to move? All right. Well, not all parts of the plants will produce sugars, but every part of the plant needs sugars. Right? Sugars are providing energy like a food source for these plants. So we would say that sugars move from sources to sinks. Right? A source is any organ that produces more sugars than they require. So typically this is only going to be leaves, though sometimes if we're talking about plants that have photosynthetic stems, this could include the stems as well. Right? Any organ that is actively photosynthesizing produces sugars. Right? And they're overproducing. They have more than what they need, so they're going to send those sugars to the sinks. All right, so sinks are organs of the flowers that do not photosynthesize. They are not producing their own sugars, but they still need the sugars. Right? So we're going to move the sugars from the sources to the sinks. The sinks might be areas like the roots, right, or the flowers, or even new fruits that are developing on the plants, or new growths that can't quite sustain themselves. They're not producing enough sugars to sustain themselves yet. So if we look at the pathways of translocation, I just want to mention the specific cells that actually produce these types of sugars. Right? And if we look at this image here, this is the cross-section of a leaf. Right? Inside of the leaf, we have these cells that are called mesophyll cells. And so these mesophyll cells can be divided even further into two different categories: the palisade mesophyll or the spongy mesophyll. Right? They're both going to function in the same way. They're both part of photosynthesis. Right? They have chloroplasts. They're able to make sugars. Right? The difference being in how they capture light. We'll go into this a little bit more in depth next week. The palisade mesophills will capture the light first, and then the spongy mesophyll kind of gets the leftover light. Right? So in this area of the leaf, we are actively producing sugars. Right? The sugars will then leave these cells, enter into the veins, enter into the phloem here, and then be moved throughout the plant to where they are needed. I like to think of the analogy of getting sugars to the parts that need them as like acquiring oil, and eventually it ends up in your gas can. Right? So we need the initial energy acquisition. We need to drill for oil. Right? Just like the leaves of plants need to make the sugar, right? We then need to move the sugar, and we move that through our phloem, just like oil is often transported through pipelines. And finally, it's going to reach the area where it's actually going to be consumed, right? So for the oil, that might be the gas pump. It might be pumping gas into your car. For plants, that's going to be other areas of the plant that need these sugars, like fruits, like roots, like new growths. Now, it's important to note that not all sources supply all sinks. Right? So there's a couple of different determinants as to which source will supply which sink. Proximity is one of them. Right? Sources are typically going to be close to the sinks that they serve. We can see in this image here that the sink in this case is this newly growing part of the plant, right? These newer leaves, these leaves, well, yes, they're photosynthesizing. They're just not photosynthesizing enough to keep up with the sugar demand because they're actively growing, right? They're new growths. They're not mature parts of the plant yet. The leaves closest to those actively growing areas will be the ones that supply the sugars to this particular sink. We can also think of development of the plant. Right? At certain times of the year, we might need to aliquot out more sugars to different parts of the plant than in others. Think about the times of the years where the plant might be flowering. Right? Producing flowers needs a lot of sugars. Right? Those flowers might turn into fruits. Those fruits need a lot of sugars. Right? So definitely during times of reproduction, right, the sinks of flowers and fruits might be prioritized. And then finally, simply vascular connections. Right? Who is connected to who? Right? So do they have a direct connection to a sink? If the answer is yes, they're likely going to supply that sink. Now, what is actually transported in the phloem? Well, water is actually the most abundant, right? Water is kind of the medium in which a lot of these molecules are going to be transported, right? Water is really good at dissolving components, right? So water is most abundant, and then we have everything else that's dissolved in the water. These would be things like carbohydrates, right? So sugars are carbohydrates. It would be things like amino acids and proteins. Right? So building blocks and proteins that might help regulate other functions on the plants. It might be things like hormones, right? So plants have hormones. Hormones need to be transported to where they're needed, as well as many of the inorganic ions that we often find. So many of the nutrients that maybe the plant has taken up from the soil, right? Together, the water and the solutes, we would call that the phloem sap. Right? It's the aqueous solution, so the water solution that flows through our sugar-conducting cells. In this image here, you can see that there are a lot of things that can be dissolved and be a part of the phloem sap here, but sugar is overwhelmingly going to be the majority of what we find in the phloem sap. All right, we went over these questions in class. I encourage you to pause the video and go over these questions now. All right, hopefully you have paused the video there. We talked about what term describes movement of sugars through the phloem. Well, that would be translocation. And what is an example of a source? It might be areas that are photosynthesizing, so primarily leaves. And then sink is anything that is not photosynthesizing, like the roots, some stems, flowers, fruits, new growths. All right, we're going to shift our topic of conversation here to talk about fungi, right? So fungi, they're not plants, right? But they can help out plants, all right? They often have this mutualistic relationship with plants, right? So the fungi that have these mutualistic relationships, we would call mycorrhizae. You can think of mycorrhizae as kind of like the fungus root, right? It's different than a plant root. It's basically any part of the fungus that is not part of their reproductive body, right? So it's not like a mushroom, but rather the different strands of fungus that you might see. So this mutualistic relationship, right, between fungi and plants, it's a really common relationship. It's found in like 85% of plant species. It's that important, right? And essentially what is happening is that the fungi are surrounding or growing inside of the roots of the plants, right? The fungi are really good at seeking out nutrients, right? Especially phosphorus, right? Phosphorus is often a limiting nutrient. And the fungi will give the plants phosphorus in exchange for some of the sugars that the plants have made through photosynthesis. All right, so they get to trade resources with one another. Right? So essentially, we can think of the mycorrhizae as an extension of the plant's roots. So it increases the effective root length like 100-fold or more. Right? It's vast, it's extensive, right? And really helps to increase the nutrient uptake rates. In this image we see here, we have two plants that have not been given any mycorrhizae and two plants that have been given mycorrhizae. And you can see those that have that association with the fungi grow much more than those that do not have the fungi. When we think about mycorrhizae, all right, we can classify them into two categories. One is ectomycorrhizae. So those are going to be mycorrhizae that grow on the root's surface, right? So they kind of surround the root. We can see that in this image here. The fungi here is like all this white net that's surrounding our root. The other type is endomycorrhizae, right? So endomycorrhizae can actually grow into the root and grow between the cells. All right, so let's look at these further. Ectomycorrhizae, right, are mycorrhizae, like I said, that cover the root's surface. We can see here we have the fungal hyphae in purple, right? The fungal hyphae are all these different strands, long strands of fungus. They are totally covering the surface of the root. Sometimes they can grow inside of the root a little bit, right? They'll grow in between the cells and in between the cell walls, so the spaces between the cells, but they're never going to grow into the cell walls themselves. All right, so remember, the cell walls are outside of the plasma membrane, kind of providing an extra layer of protection and rigidity to our cells. Right? Often we see the roots completely covered with the fungal hyphae. We can say that it's forming what's called this Hartig net. Right? It's a complete covering of those roots. We can also have, like I said, endomycorrhizae. So these mycorrhizae, they will grow around the root, but they will also penetrate into the roots themselves. Right? Now, they do not grow into the cell. All right? They are never going to penetrate the plasma membrane. Right? But they will grow into the cell walls of a lot of the root cells. And they can form structures that are like clumps of fungal hyphae called arbuscules and vesicles. Right? We can see some clumps of fungi here. All right, we can also see some clumps of fungi in this nice image as well. All right, fungi are not the only organisms that can have a beneficial relationship with plants, right? There are other materials like soil bacteria that can help the plants obtain nutrients. Now, if we look at the atmosphere, right, and all the gases that we might find, we can see in our little pie chart here of all the gases in the atmosphere that nitrogen is really abundant, right? So it might seem that it should be easy for plants to take up, but the problem is that nitrogen is in this gas format, or in this gas form. It can't be used by the plant when it's a gas. It has to be fixed or transferred to another molecule, essentially, to be able to be used by the plant. And so there's a couple of different ways that nitrogen can be fixed. One is actually by lightning. All right, lightning can fix nitrogen. It's really cool that lightning can do this, right? But it's not super useful because lightning is sparse, right? The other way that nitrogen can be fixed is through bacteria. And bacteria that live in the soil, they can fix nitrogen on their own, or they can fix nitrogen when they're associated with the roots of a plant, right? So when I keep saying it's going to be fixing nitrogen, right? So we're taking it from a gas and fixing it into another molecule, right? So N2 is the gas form, right? Oftentimes we're going to use an enzyme called nitrogenase, right? As well as a lot of energy, right? 18 ATP. ATP is the energy molecule in this case. That's a lot of energy, right? In order to transform this gas form of nitrogen into another molecule that plants can actually use. Now there's a lot of nitrogen fixing plants. Oftentimes we see them in legumes, right? And the nitrogen fixers or the bacteria themselves will be rhizobia. So legumes are like in the pea family. We often see them in actinorhizal plants, right? The bacteria here would be a species or a genus called Frankia, as well as, I pressed the wrong button, as well as other species of bacteria like cyanobacteria can often fix nitrogen. I don't expect you to know the names of the different nitrogen fixers here. I just want to give you some examples of what is out there. All right, I did do another couple of thought questions here. So again, I encourage you to pause the video and think about these yourself. All right, hopefully you did pause the video there and thought about them. We talked about how might mutualists be affected by changes in soil nutrient availability, right? Well, often you can think of it as like an exchange of resources, right? And if there's a lot of soil nutrients available to the plant, right, the plant is gonna use the nutrients on the soil, right? Why spend sugars to the fungi to get nutrients when they can get the nutrients themselves, right? So the mutualists might end up having less sugars given to them. Likewise, if there's too little soil nutrient availability, the plants might become more reliant on those mutualists. How might mutualists be affected by decreases in photosynthetic activity? Well, if we're decreasing in photosynthetic activity, right, the plants are making fewer sugars. So that's fewer sugars that they can give away in exchange for nutrients from these mutualists, right? So these mutualists are just not going to get as much sugars, right, because they're going to hang on to their nutrients. They don't want to give away their phosphorus and nitrogen for nothing. All right, the last little bit of the lecture here, we went over NASA and how they are doing fungal research. One of the cool ways that NASA is doing research here on Earth is actually through satellites up in space. So they're using satellites to actually detect the presence of mycorrhizae, Right? Now, in the paper that was published that gave this information here, they were really looking at mycorrhizae in forests. And if we look at forests, they have this really specific spectral signature, right? So we send out wavelengths of light. They're going to give us very specific wavelength signatures back to us. Now, researchers looked at these forests and tried to look for patterns in these signatures that were being sent back. Right? And they were trying to associate those patterns with the microbial networks that are found on Earth. Right? So they compared the patterns that they found with the known fungal networks on Earth, and they're actually fairly accurate. Right? They're 77% accurate. And there's still ongoing research to further this along. All right, well, but 77% is pretty incredible. All right, we are looking at underground, right, very tiny organisms, right, these very slender hyphae of fungi, right, again, underground by the use of satellites. So pretty cool ongoing research. We're also sending fungi in space because fungi could actually be really useful. So first of all, some fungi are just not affected by radiation, right? That's crazy, right? Most organisms on Earth will be affected by radiation, and it's actually affected in a very harmful way, right? Too much radiation can cause consequences for your DNA. It can cause cancers. Some fungi are they're simply not affected. Other fungi will actually use radiation. Right? And this is an ongoing area of research, right? And we hypothesize that these fungi are using radiation as their source of food, which is crazy, right? We call this radiosynthesis. It's still being researched. Fungi that do this are often or can be found around like the Chernobyl reactor site. Crazy, right? If you want to, you can watch this video I have linked on the slides here. To learn a little bit more about how this happens. Let's see. Fungi in space, right? If we're thinking about maybe eventually colonizing other planets or going to other planets for long term, the moon, the Mars, or Mars, we don't have a lot of nutrients in those regolith soils, right? And mycorrhizae, they're really, really great at getting nutrients. So we might think about mycorrhizae as a way to help plant growth. Mycorrhizae could also be used in building materials, right? Fungi, they grow fast, right? We don't have to ship building materials. We can simply grow our building materials. It's kind of crazy. So I'd encourage you to watch this video as well as the video I have linked onto the next slide here, okay? But I'm going to skip to my last slide and talk about some research that has been done on mycorrhizae and how well they might fare in microgravity conditions, right? And it's been found that microgravity negatively affects mycorrhizae, and negatively, because they're negatively affecting the mycorrhizae, lower the amount of plant phosphate uptake. And we can see in this image here, image D is the mycorrhizae grown in microgravity compared to regular gravity in image C. And we can see there's a difference in growth. It looks stunted, right? But overall, the researchers concluded that even though mycorrhizae is not as effective in growing in microgravity, it's still going to be able to support plant biomass production in microgravity, right? So we're still saying that even though there might be some negative effects, they're still going to play a really important role when we think about the future of plants in space. All right, and then we have this homework here. I also went over reading a scientific paper in class. I have a video that was made last year, linked on Moodle, that you can watch through as well.